Okay, so in this video, we will discuss on the secondary growth in the normal roots. So we had already talked about this, the growth, the TS section, as you can see in this figure, that is the anatomy of the normal roots. So now we will take a look at the secondary growth. Now, secondary growth in the Cycas root, it occurs in the same manner as in case of dicot angiospermous roots. It initiates in the very beginning when the root is still young. Here, a few parenchymatous cells present in between the xylem and the phloem. Alright, when the root is young, these the parenchymatous cells which are present in between the xylem and the phloem, they become meristematic. Okay, they become meristematic. Meristematic cells, you know, they are those actively dividing cells, okay, which form new tissues. So these what happened at the beginning, how it initiates, the few parenchymatous cells which are present in between the xylem and the phloem, surrounding the phloem, they become meristematic and they will start functioning as a cambium. Now, also you know that a cambium is a plant tissue which will form phloem, xylem or cork. Okay, where the phloem, xylem or cork, they grow by division. So these parenchymatous cells, they will start becoming meristematic, and also they will start functioning as a cambium. Alright, so these cells here, they will start functioning as cambium. Now, just take a look at it. This diagram, see here, you can see this portion here. This is the cambium. Alright, so this is the cambium. These cells, they start functioning as a cambium, and the cambium strips, you can see these are the cambium strips. All right, so these cambium strips, they usually look crescent shaped. Okay, just like a curve shape, just like a little silver of moon. Okay, moon shape, crescent shape of moon. And these cells of cambium, they start cell division. All right, they will start cell division, and they will produce xylem towards the center. So the xylem will start forming towards the center of the pith, that is known as centripetally. Whereas the secondary phloem, all right, just take a look at this one here. Okay, this is the secondary phloem. The secondary phloem will start forming towards the periphery, that is centrifugally. So due to the formation of the cambium, the cells of the cambium, they will start cell division. I'll just repeat whatever I said just now. They will start cell division and produce xylem towards the center, that is centripetally, and secondary phloem towards the secondary xylem towards the periphery. Now, besides the secondary xylem and secondary phloem that is formed right in, as you can see in this figure C, the cambium cuts broad parenchymatous rays. Okay, so besides these secondary xylem and secondary phloem which is forming, this cambium, it also cuts the you can see here, all right, this xylem, this cambium, it also cuts a broad parenchymatous rays. Thus the cambium, we say that it produces monoxylic secondary wood similar to the stem. Now, monoxylic meaning the non-compact wood. Okay, the non-compact wood with a large amount of parenchyma, large pith, and as well as cortex. That means less amount of xylem, tracheids or wood, because we also have polyxylic wood. So here monoxylic secondary wood, which is similar to the stem of the cycas, is formed, okay, due to the division of the cambium. The cambium, it cuts a broad parenchymatous rays, forming the monoxylic wood. In the meantime, here in the pericycle, all right, as you can see this layer here just below the endodermis, it is the pericycle. So in the meantime, what happened in the cells of the inner layer of pericycle lying behind the protoxylem, all right, so the cells here in the pericycle which lie behind these protoxylem, they also will become meristematic, that is become actively dividing cells, and they will start behaving as cambium strips, okay. So when they start behaving as cambium strips, they will join the crescent shaped cambium, okay. They will join the crescent shaped cambium strips, and these cells, they produce living cells of primary medullary rays opposite the protoxylem. So just remember that the cells of the pericycle which is lying behind the protoxylem, they will also behave as cambium, joining the crescent-shaped cambium strips, and they will produce living cells of primary medullary rays opposite the protoxylem points, okay. So opposite the protoxylem points, here they will form the primary medullary rays. And what happened, I will show you in this diagram. Simultaneously, after the formation of the cambium strips, the secondary phloem and the secondary xylem, so what happens simultaneously, the cork cambium, here, which is also known as phelogen, it develops in the outermost layer of the cortex. Alright, so the outermost layer of the cortex just below the endodermis will lead to the formation of a cork cambium known as the phelogen. And this phelogen, it produces dead cells of cork. You can see here, these are the dead cells of cork on the outer side, known as the phloem, and the living cells of secondary cortex, okay. These are the living cells of the secondary cortex towards the inner side. So just remember, towards the outer side, the phloem or the cork is present, and towards the inner side, the living cells of secondary cortex or pheloderm is present. And sometimes another cork cambium layer, it will also develop here in the pericycle. So sometimes in the pericycle, the another layer of cambium will develop, and this cambium, it will cut cork towards the outer side, and secondary cortex towards the inner side. So the cork cambium and secondary cortex together, they constitute what we call periderm. But what happened, this periderm produced by the inner phelogen, it takes a superficial position in the root as, the cork cambium and the cortex, okay, they will take a superficial position in the root. Why? Because this outer periderm here, remember periderm, cork cambium and cortex, they constitute the periderm. This periderm is short-lived and it will be soon sloughed off. So that is why this periderm produced by the inner phelogen, it will become superficial in position. And what happened later on, the lenticels, okay, that is the cells which are formed in the stem of a woody plant, which allow gas exchange between the atmosphere and the internal tissue. Okay, that is the function of the lenticels. These lenticels, they will develop in the old roots. So this is the secondary growth which occur in the normal root of Cycas plant.